So welcome to this series of 4 lectures on protein misfolding, its association with disease and therapeutic approaches to treatment. Correct folding, as shown on the right, is required for proteins to function. And on the left is what can happen when proteins fail to do so. They can misfold and stick together to form structures known as amyloid fibrils that are toxic to the cell. And this is a dramatic example of this phenomenon. This is lysozyme amyloidosis, a disease in which literally kilograms of the protein lysozyme accumulate as clumps known as fibrils in the patient's organs, including the GI tract, liver, and kidneys. So far in the part 2 lectures, you've heard about some of the most common classes of drug targets, for example, GPCRs in Professor Davenport's lectures. These proteins are good examples of what is sometimes referred to as the druggable proteome, which is why they're the most common drug targets. But of course, that doesn't mean that there aren't many other targets that are equally important. And these are what you'll be hearing about in my lectures. In protein misfolding diseases, there have actually been some remarkable drug discovery success stories. For example, cystic fibrosis, which you'll have heard about from Dr. McVinish's lectures, but also some very high profile problems and controversies, including drugs for Alzheimer's and other neurodegenerative diseases. What many protein folding diseases have in common is that they're hard to drug. This slide illustrates the scale of the problem. The total number of proteins encoded by the human genome is around 30,000. And the number of proteins that are readily druggable on the left-hand side of this image is around 3,000. And I'll explain what we mean by druggable in the next slide. But also the number of proteins that need drugging on the right-hand side of this image is also estimated to be around 3,000. And the intersection between the two gives an estimate of the number of exploitable drug targets that there are today. So you can see that the majority of the proteins that we need to drug remain, so these proteins here remain undrugged. And this figure is from a review from 2002, but estimates are very similar today. So not much has changed in the intervening time. So what do we mean by druggable proteins? Well, most belong to a narrow range of protein classes, and that's shown here in the bottom right. You can see in these charts of successfully drugged protein families that there are enzymes, for example, kinases, there are receptors like GPCRs, there are ion channels. So by druggable, we mean proteins that have natural small molecule ligands from which small molecule drugs can be designed, or for which it's been straightforward to obtain good antibodies. And the quote at the bottom here was from this follow-up review in 2017, over a decade after the original one, that says essentially we're still going after the same class of drugs. So of the 361 new molecular entities approved by the FDA over a 10-year period, only 6% targeted a previously undrugged protein or protein domain. So back to the protein misfolding diseases, you might be familiar with the fact that protein misfolding and aggregation is implicated in neurodegenerative disorders such as Alzheimer's and Parkinson's. Less well known is that many other diseases are also caused by protein misfolding, and some examples are given in this table. So for each disease, I've listed the protein that misfolds and whether this causes loss of the normal function or gain of protein function. Just hold on one sec. Let me just check that I'm recording. Yes, I'm recording. Okay. So examples of, apart from neurodegenerative diseases, which are the obvious ones, are cancers, where mutations cause impaired folding of tumor suppressor proteins like P53 and BRCA1. And diseases like cystic fibrosis that you've heard about from Leslie McVinish's lectures, and metabolic disorders at the bottom here, An impaired folding of certain GPCRs is involved in diabetes and retinitis pigmentosa. And also protein folding mutations are associated with cataracts and thyroid dysfunction. So a very wide range of very different diseases. And so why is protein folding or misfolding such a problem? Well, even in healthy cells, it's estimated that up to 50% of synthesized proteins fail to fold. And as well as being hugely costly in terms of the amount of the cell's resources that are spent on protein synthesis, the other problem is the cell has to get rid of these misfolded proteins. And how do cells deal with that? Well, there are various cellular mechanisms that I'm going to talk about today. And some of these mechanisms fail in disease states. And drugs have been developed to target those particular mechanisms of protein quality control and protein misfolding. So these are the learning objectives of the lecture series. Firstly, to understand how many diseases have their pathologies in protein misfolding, and that this provides an opportunity for therapeutic intervention. In lecture 1, we'll briefly review the basic principles of protein folding and how protein folding in the cell is coordinated by the cellular machinery. The basics are important because both drug discovery and drug optimization for protein misfolding diseases have required an understanding of the underlying mechanisms of folding. We'll cover how the cell's quality control machineries, the so-called proteostasis network, helps in protein folding and identifies abnormal and damaged proteins and ensures that they're efficiently cleared from the cell. We'll discuss the errors in protein folding that lead to disease and how quality control machineries can become impaired in disease states. And the lectures will illustrate how researchers have gone from the bench to the bedside, translating what's known about the molecular pathologies of protein misfolding disease to develop new treatments. And we'll discuss some of the key success stories as well as ongoing challenges in this area of drug discovery. So useful papers can be found on the Moodle pages for each lecture. And the key ones are starred in the slides and the handouts. I've put up supervision slots over the next couple of weeks. And to use this time most effectively, I recommend you attempt either one or both of the essays here or essays from past papers. And also do send me them ahead of the supervisions. I'll endeavour to mark them if you send me them a few days before the supervisions, and I'll email them back to you with comments. So protein folding is the last part of the genetic code that translates the DNA sequence into the functional protein. We want to understand why do different sequences fold into different structures, and also how are they able to avoid misfolding. The landmark experiment that started the field over 50 years ago was performed by Amundsen and won him the Nobel Prize in 1973. He took the protein RNase A and unfolded it by adding chemical denaturant, urea, and the reducing agent beta-mercaptoethanol. He then removed both by dialysis, and he showed that the protein spontaneously refolded to its native functional form. No cofactors were needed, and this means that all the information required to specify the 3D structure is encoded in the amino acid sequence. So the protein folding code can be divided into two parts. The first has to do with thermodynamics. Why is the native state the most stable state of the protein? And second has to do with kinetics. How do proteins fold rapidly and on a physiologically relevant timescale? This is the simplest situation in which the protein can only exist in two forms, either the fully unfolded state or the fully folded state. And the stability difference between the two is the free energy of unfolding. And why is the folded state the most stable state? Why is it more stable than the unfolded state? Well, it's a combination of different factors. The two states have very different properties, as summarized in the table, and this is what drives folding. So the unfolded state is it is expanded, it has a large solvency accessible area. The folded state is the opposite. The unfolded state is very disordered, and actually the loss of entropy when a protein folds is unfavorable. But that's overcome by all the favorable interactions that the protein makes within the folded structure. So most proteins, small proteins, so under 150 amino acids, as I said, can only exist in one of two states, either fully folded or fully unfolded. And in other words, the protein structures are highly cooperative. And what we mean by this is that all the interactions are interdependent on each other. So breaking a few bonds destabilizes all the other bonds too. So one part of the structure can't unfold without the rest of the structure unfolding, and partly folded structures don't form. And cooperativity is very important in preventing misfolding, because protein misfolding often starts from a partly folded state. Large proteins, however, multi-domain proteins, for example, as are commonly found in eukaryotic cells, have less cooperative structures. So they can adopt partly folded states, and that means that they're more susceptible to misfolding. The other property the protein needs to have, as well as thermodynamic stability, is kinetic stability. So there needs to be a high energy barrier separating the folded state from the unfolded state, so that requires a lot of energy for the protein to unfold. So the second part of the protein folding problem has to do with kinetics. How do proteins fold on a physiologically relevant timescale? And in the 60s, Leventhal posed this paradox. If we consider a protein of 100 amino acids, if each amino acid could adopt just 2 different conformations, then there would be 2 to the power of 100 possible conformations of the polypeptide chain. And this means that if the protein folds via a random search of all these conformations, it would take 4 times 10 to the 8 years for the protein to fold. But small proteins can fold on a very rapid timescale, even milliseconds to minutes. So how do they do this? And it was thought that the solution must be folding pathways that guide the protein, involving a series of partly folded intermediate states. So researchers started trying to map these folding pathways. And 2 approaches were developed in the 80s and 90s that allowed us to characterize folding pathways at very high resolution. The first was pioneered by Alan First here in Cambridge. And this was taking advantage of the advent of modern molecular biology techniques in the 90s, which meant that one could clone any protein. And it wasn't possible to do that previously. It seems amazing now. So you could make any protein, and you could make mutations at individual sites. And this site-directed mutagenesis approach allowed us to map protein folding pathways in detail. And this paper here was a landmark in the field. At the same time, in silico methods, so molecular dynamic simulations, also started to transform the field. These simulations allowed us to follow protein folding at even higher resolution. So these are images from simulations of protein unfolding. A number of different proteins are shown here, where we can follow every atom of the protein and obtain high resolution snapshots of how the structure changes at key stages along the unfolding pathway. It's harder, i.e. it's heavier on computer time to simulate folding than unfolding. And that's because folding starts with many conformations in the unfolded state, whereas there's a single folded structure that's the starting state for unfolding simulations. So folding simulations were very difficult to do when computers weren't very powerful. They can now be done for very small proteins. And on the left, you can see, so this is a small protein. This is a video of a simulation of its folding. On the right, there's a little black dot here at the top. This is an energy surface for a protein, starting at the lots of unfolded states of high energy at the top and going to the unique folded state, the most stable state at the bottom. And you can see when I show you the simulation, this black dot moves, and it moves into this misfolded or partly folded state in the middle of the simulation, from which it then has to get out of to be able to fold properly. So you can see here the protein is starting to fold up, it's still sampling unfolded conformations, and then at some point it falls into this kinetic trap, this partly folded state, from which it then has to extract itself to fold properly. So this was quite an achievement. This is a small protein, but folding a small protein like that in silico requires a huge amount of computer power. And for longer simulations, this group in the States, Vijay Pandey's group and colleagues, set up what's known as folding at home. So this harnesses the power of thousands of home computers to run large simulations in the background while people are working or sleeping. And this was really revolutionary. In 2000, they launched their software, people could run them on their PCs, and this massively enhanced the computer power that was available. So far, this all sounds a bit esoteric, but now we see that these computational methods, so simulations of the dynamics of protein structures, how proteins move and how that relates to their function, can be applied to aid in drug discovery and development. And these two papers describe some of the findings obtained by the folding at home volunteers. The first one at the top is simulations of a GPCR that revealed the fluctuations, so how the protein moves between its inactive and active states. And these simulations helped explain the effects of agonist drugs. The second is a simulation of a set of simulations of the COVID-19 virus proteome. And this involved over 1 million folding at home volunteers. And this work uncovered the mechanisms of conformational changes that are essential for the replication cycle of the virus, and also revealed a number of potential therapeutic opportunities that had previously not been observed in crystal structures, including hidden antibody binding sites. On the right, you can see a graph showing how the pandemic escalated, and also, so that's in blue, the number of COVID-19 cases, and in orange, you can see how the number of folding at home volunteers increased in parallel. So Sony added folding at home to its PS3 platform in 2007 and gained a very large number of users. So all you had to do was download the software and leave the PS3 on while you weren't gaming. And the results was that literally millions of both PS3 and PCs collectively achieved performances that the top supercomputers could not equal. The numbers went down when PS3 stopped running it, but then last year everything changed. So folding at home went suddenly, very suddenly, from 30,000 volunteers in February of last year to 400,000 in March. In part, this was because of this tweet that asked the public to donate their computing power to help find therapies for COVID-19. Another 300,000 users came on board after that, and subsequently there were so many users from all around the world, as you can see in this figure here, that the database ran out of simulations for them to crunch, and the data coming in was so great that the servers were overloaded. And actually, at its peak performance, folding at home was 100 times faster than the 100 fastest supercomputers combined. And the top 10 of these are compared in this bottom figure here with folding at home. So you can see folding at home is more than 10 times more powerful than the next most powerful supercomputer. So this is really promises to revolutionize our understanding of how drugs work and drug discovery and development. So-called citizen scientists are even more actively involved in designing new proteins. So Fold It is a computer game in which players can do just that. For various reasons, it turns out that these amateur scientists are even better at some aspects of protein design than the protein design algorithms developed by professional scientists. And a number of papers have come out detailing the results of these design efforts. This one in Nature in 2019 had literally tens of thousands of authors, all of them folded players. Earlier this year, protein folding again hit the headlines. So an AI program known as AlphaFold 2, developed by the company DeepMind, claimed to have effectively cracked the protein folding problem. Both Foldit and AlphaFold work by training computers on the database of known protein structures so as to learn the underlying rules and thereby be able to predict the structures that any given polypeptide sequence will fold into. So who is DeepMind? It was founded by UCL scientists, including this guy at the bottom, Demis Hassabis, and it was acquired by Google in 2014. And they started by using their AI approach to play the chess-like game of Go, beating the world champion at it, before moving on to the strategy game StarCraft, and then into the academic field of protein structure prediction and this protein structure prediction competition known as CASP. So CASP stands for Critical Assessment of Structure Prediction. It's a worldwide protein structure prediction competition that runs every 2 years, where academic groups compete to predict structures of a set of target polypeptide sequences. It was started in 1994 and the results were pretty good. So the academic groups involved correctly predicted around 60% of the protein structures. But when AlphaFold entered the competition in 2019, so that's here, this is AlphaFold 1, the number jumped to almost 75%. And then 2 years later, the improved AlphaFold 2 won the competition with the success rate shooting up to 90%. And the protein structure prediction capability provided by AlphaFold has really big implications for drug discovery. Although there are limitations, of course, it's not so good at some types of structures, and it can't help us with a very large number of proteins that are intrinsically disordered, and I'll come on to those later. So now computer simulations of proteins and of their interactions with small molecules is becoming a really integral component of drug discovery toolkits, both in big pharma as well as in academic labs. And these are some examples here, and this is a recent review article in Nature. In April of this year, the biotech company Evotec announced a phase 1 clinical trial on a new anti-cancer molecule created in collaboration with this company here, Exscientia. That's an Oxford company, originally a spin out from the University of Dundee, that applies AI techniques to small molecule drug discovery. And it would have taken the conventional drug discovery process several years to find the drug candidate, in this case, an A2A receptor antagonist for solid tumors. Instead, it was found in 8 months by harnessing Exscientia's AI design platform. So this platform can computationally sort through and compare the properties of millions of small molecules, looking for 10 or 20 to synthesize and test and optimize experimentally before selecting the eventual drug candidate for clinical trials. And within 3 weeks of this, Exscientia announced a $225 million financing round. The company did the same last year, announcing a drug candidate to treat OCD. This and the oncology drug are the first 2 molecules designed with the help of AI to enter clinical trials. And it's almost certain that there will be many, many more to follow. So back to protein folding. We know that proteins can fold to their native state in vitro, but in the cell, many proteins need help to fold spontaneously and rapidly, and also help to avoid protein misfolding. This image illustrates the 3D, the 3D nature of the problem, and also the third layer of the protein folding code. We have the primary structure. We have, so that's the amino acid sequence. We have the secondary and tertiary and quaternary structures. And last is the quinary structure. And this is a term that's been used to refer to the behavior of the protein in the cellular context. And as an illustration, this is a short simulation of the interior of an E. coli cell. Oops. So the video is in slow motion, slowed down by a million times. You can see that the proteins need to fold and stay folded when they're constantly being jostled around in this very highly crowded, very dynamic environment. So how does this go wrong in disease, and how can we drug these pathologies? So the protein has to get from the denatured state to the native state here in the cell without misfolding. And the danger is if it adopts an intermediate state, for example here, and this state will have exposed hydrophobics that would normally be buried in the native state of the protein. These are sticky and can clump together and to form beta sheet aggregates that can further organize to form these very, very stable and potentially toxic amyloid fibrils that accumulate in disorders such as neurodegenerative diseases. And these processes are often irreversible. So the protein, once it's in an intermediate or a denatured state, forms these aggregates that are reversible, but these fibrillar aggregates are unreversible. So once it's there, it can't be reversed. This is an energy diagram with different states of a protein along the x-axis and their energies along the y-axis. And in order to understand folding in the cell, as well as the productive folding events on the left here, we also need to know the other half of the energy landscape. We need to understand misfolded states and aggregated states, understand the fact that they're very stable and very hard or impossible to reverse. So misfolding and aggregation in the cell are prevented by interactions with molecular chaperones, and slow folding steps, for example, disulfide bond formation, are accelerated by another class of protein known as folding catalysts. And it's important to note that both chaperones and catalysts are found in all compartments of the cell, so the nucleus, the ER, mitochondria, cytoplasm, so all compartments in which proteins fold. So Ron Lasky, a Cambridge professor, came up with the term molecular chaperones in the late 70s. He defined them as a group of unrelated classes of proteins that mediate the correct assembly of other proteins, but aren't themselves components of the final folded structure. And chaperones work by binding and stabilizing otherwise unstable states of the protein. And by binding and releasing the protein multiple times, that facilitates its correct folding. And also the attainment of its final state, so that as well as folding, that could be assembly of different subdomains and subunits, transport to different subcellular compartments, switching between active and inactive conformations, and targeting for disposal by degradation. So all of these processes in the cell are facilitated by molecular chaperones. And although there are a very wide variety of chaperones, they do share some common features. As I said, they prevent incorrect interactions within proteins and between proteins, so just like human chaperones. They don't increase the rate of folding, but they do increase the yield of folded protein. Many are induced or upregulated under stress, so for example, high temperatures, and hence many chaperones are often referred to as heat shock proteins. Many chaperones recognize exposed hydrophobic clusters on their substrates. So these are in substrates that are partly folded or misfolded. They have hydrophobic residues that would normally be buried in the folded protein. And lastly, many use ATP hydrolysis as a fuel to elicit changes in conformation of the substrates and that ultimately drive their folding. Chaperones are also part of the cell's quality control machinery that detects misfolded proteins and target them for degradation. And we'll come on to this in lecture 2. And it's been shown that over-efficient quality control machinery can cause diseases, and I'll cover this in lecture 3. Deregulated quality control can also be exploited by the cell for uncontrolled proliferation in cancers. Again, this will be covered in lecture 3. And lastly, failure of the quality control machining with disease, with age, can lead to a range of disorders, including most notably neurodegenerative diseases, and I'll cover these in lecture 4. So the sequential chaperoning and quality control processes required during a protein's life are shown schematically here, and they're discussed in this very nice review by Judith Friedman and colleagues. In the next slides, in the first part of lecture 2, we'll go through these different stages of a protein's life cycle, of its biogenesis. The key players, the chaperones, and the protein degradation machinery are the backdrop to the therapeutic interventions that we'll discuss in the second part of lecture 2 and in lectures 3 and 4. So the first part of these processes is co-translational folding. So protein folding can start during synthesis on the ribosome, and this is very different from protein folding in the test tube. Firstly, because the N-terminal residues of a polypeptide are available for folding before the C-terminal residues have been synthesized. So in other words, not all the information that's encoded in the amino acid sequence is available. Second, the polypeptide chain is conformationally constrained by the ribosome, so the C-terminal 30 amino acids or so are attached, and they're combined within the ribosome exit tunnel. Also, the newly synthesized unfolded chain emerges into the very crowded and dynamic environment of the cytoplasm, where aggregation can very readily occur, so it needs to be protected, and I'll come on to this later in the lecture. And third, the rate at which translation occurs will affect the mechanism of cellular folding. This is something that was appreciated many years ago, but really in the last 10 years has been characterized at a much higher level of detail. So translation rates are slower in eukaryotes than prokaryotes, and that's because these slower rates will facilitate folding in multi-domain proteins, which are obviously more common in eukaryotes. And we'll come on to this next. We'll see that the translation rates are not constant within a polypeptide chain, so they can slow down or speed up depending on the mRNA sequence. So this is a sort of paradigm difference between folding in the test tube, where it's just the amino acid sequence that affects the folding reaction and the final folding state. In the cell, the mRNA sequence as well as the protein sequence affects the folding process. So the impact of translation on in vivo folding is illustrated here. Codons are optimized both to coordinate correct folding and also to prevent misfolding and aggregation. And one aspect is ensuring translational fidelity. So this is important because proteins are only marginally stable in the cell, and errors in protein synthesis result in around 1 misincorporated amino acid per 10,000. And this could tip the balance from correct folding to misfolding. So there needs to be a very high level of translational fidelity. Second, the role of the ribosome is to coordinate the translation process. So most amino acids are encoded by several synonymous codons that are under selection for translational speed. Translationally optimal codons are recognized by tRNAs with higher availability and are thus translated more rapidly. They also confer, as a result, higher translational accuracy. And these optimal codons tend to be associated with evolutionary conserved sites in the protein, i.e. functionally important sites, and also with sites that are structurally sensitive or aggregation prone. So sites at which translational errors are more likely to disrupt folding. So selection for optimal codons is an important determinant of coding sequence evolution. But it turns out that non-optimal codons are also conserved, underlining their importance for correct folding in the cell. So these non-optimal codons are recognized by low availability tRNAs, which means that translation is slower. Their positioning along the mRNA coding sequence is thought to allow the nascent chain more time to begin folding co-translationally. So what we find is that there are patterns of optimal and non-optimal codons that correlate with the secondary structure of the encoded protein. Structures that can fold co-translationally, for example, helices, as shown here. So the helix can fold within this very narrow ribosome exit tunnel, do so co-translationally and tend to be encoded by non-optimal codons, whereas beta sheets, like shown here, can't fit in this exit tunnel, and they tend to be encoded by optimal codons. So there's this orchestration of the rate of translation that correlates with the structure of the protein and its ability to fold, and also its tendency to misfold. You might wonder how co-translational folding is related to disease. Well, this is an extreme example of how mRNA sequence and consequent translational kinetics affect folding and cause disease. So this paper was published in 2007, but there have been many other examples found since. These also showed that a single SNP that changed a codon from an optimal to a non-optimal one changed the co-translational folding of this MDR protein. So instead of folding to its correct structure, it folded to a different non-functional structure. So an apparently silent SNP, i.e. a SNP that changed, that didn't alter the protein sequence, so the amino acid sequence it encoded was the same, but the mRNA was different, can still cause disease by disrupting the co-translational folding pathway so profoundly that the protein misfolds. So we've talked about co-translational folding. Proteins subsequently emerged from the ribosome into a highly hostile environment containing lots of other proteins and other macromolecules, with many opportunities to misfold. And to prevent this, folding requires the coordinated action of a series, usually of chaperones, acting both during and after translation. So a newly synthesized protein, shown here in red, will emerge from the ribosome and be met by a host of different molecular chaperones, shown in blue, some examples of which are shown in blue here. And despite their very different architectures, which you can see here, chaperones have some common mechanistic features. Firstly, in terms of substrate recognition, most chaperones have very little substrate specificity. So the question is, how do they recognize non-native, so that's partly folded or unfolded or misfolded states rather than native states? And they do so by binding to solvent-exposed clusters of hydrophobic residues that would normally be buried in the correctly folded protein. To assist in folding, most chaperones use ATP hydrolysis. So ATP binding and hydrolysis produces energy and also drives conformational changes in the chaperone that are then translated to the substrate and allow it to fold. There are, however, a small number of chaperones that don't use ATP and simply have a handover role. So they just protect the nascent chain during assembly of the protein. Trigger factor is the best characterized of the ribosome-associated chaperones that protect the nascent chain. And it consists of three different subdomains. There's an N-terminal domain for binding to the ribosome, shown in red here, a peptidyl proline isomerized domain required to catalyze the slow process of proline isomerization, and a substrate binding domain shown in blue. And this chaperone works by attaching to the ribosome. So it sits at the exit of the ribosome tunnel and forms a cavity or a cradle over that tunnel. It holds the nascent chain, shown here in orange, in the cavity, providing a protective environment to prevent aggregation while the rest of the chain is being synthesized. And it also actively unfolds any incorrectly folded structures. as I said, this protein doesn't have ATPase activity, so it doesn't use ATP hydrolysis as fuel. Instead, it's thought that it induces unfolding by presenting binding surfaces to the substrate that prevent intramolecular interactions that are non-native of the nascent chain. Another key chaperone is GROEL, or TRIC CCT in eukaryotes. GROEL is also known as HSP70. It's a heat shock protein, and it directs the folding of a very large number of proteins that are unrelated in sequence or structure, both from nascent chains and also from denatured states after cell stress. It forms this cylindrical structure, barrel-like structure, made up of 2 stacked rings, each of which have 7 subunits, and there are cavities at the ends where the substrates can enter and exit. The 3 subunits are shown here, 2 from the top ring and 1 from the bottom ring, and you can see that each subunit has 3 different subdomains shown in red, yellow, and green. The structure of GROEL and the mechanism it uses for chaperoning is shown more clearly here. So it has a co-chaperone, GROES, and substrates bind, so GROES sits on the top of the barrel. Substrates bind to hydrophobic sites, shown here, in one of the subdomains of GROEL. And binding of GROES and ATP to the GROEL ring induce large conformational changes that I think you can see here. So the GROEL goes from this structure to this extended structure. That creates a cavity that traps partly folded states in this chamber, this barrel, allows the protein to fold and prevents it from sticking to other proteins and misfolding. So substrates bind hydrophobic residues lining the cavity of GROEL. These are shown in yellow here, these little yellow bars. So this is on the left, this is GROEL before GROES binds. In the middle image, GROES and ATP binding causes this hydrophobic surface on the right to change. So this hydrophobic surface moves out of the way, and that causes the bound hydrophobic residues in the substrate, shown in these yellow dots, to fold. You can see that more clearly in the top view. So without GROES, you have the hydrophobic residues of GROEL here, aligning the cavity, hydrophobic residues of the substrate of the yellow dots. And then with GROES binding and ATP binding, those hydrophobic residues of GROEL move out of the way, and consequently the protein buries its hydrophobic residues and folds. Another key chaperone is HSP70. It has a range of different functions. As well as folding, it's also involved in assembling proteins into complexes and trafficking proteins to different organelles. And because its substrates are very specific, it's been implicated in many diseases, and it's therefore a very important drug target in a wide range of diseases, including cancers and neurodegeneration. HSP70 has 3 subdomains, an ATPase domain shown here in green, a substrate binding domain in blue, and a so-called lid domain in orange. And that sits on top of the substrate shown in yellow here. There's also a flexible linker between these domains. And the substrate binding domain has a cleft cap that's capped by this flexible lid. So the substrate sits in here. When ATP binding occurs, it triggers a conformational change in the ATPase domain. As you can see on the right here, that means that the other 2 domains, the substrate binding domain and the lid, pack against the ATPase domain, and that results in opening of the lid and release of the substrate. After ATP hydrolysis, the domains separate and the lid closes again over the substrate. So you get these cycles of binding and release of short regions of the substrate that have the net effect of unfolding non-native structures, as well as allowing the polypeptide another attempt at refolding. And HSP70 also facilitates other processes that involve folding and unfolding. So, for example, translocation of proteins across membranes during trafficking, and also unfolding of proteins and preparing them for degradation. I'll discuss that further in lecture 2. Lastly, HSP90. This chaperone has a very small number of substrates, around 5 to 10% of the human proteome. It has both stress response and housekeeping functions. It's involved in the late stages of folding, so more protein assembly of different subunits or cofactors. It's also been shown to suppress mutant phenotypes, and this can be good or bad. It can be good because it allows proteins to evolve mutations that improve their function, but that are destabilizing, so it protects proteins so that these beneficial mutations can appear. But it can be bad because harmful mutations, for example, in cancer, are enabled by HSP90. It's a promising drug target. Its substrates, known as clients, are often degraded when HSP90 is inhibited. And multiple pathways are hit by HSP90 inhibitors because of these client proteins. Inhibition of HSP90 tends to induce cell cycle arrest and trigger apoptosis. And HSP90 inhibitors have been shown to be selective for cancer cells, killing cancer cells. And that's thought to be because cancer cells require oncogenic driver proteins, so this is known as oncogenic addiction. And many of these proteins are HSP90 clients that can't fold or assemble without it. These are some of the best characterized HSP90 client proteins. So HSP90 is involved in protein activation. The first example is folding and assembly of cyclin-dependent kinases and their assembly with cyclin activating subunits. Second are steroid hormone receptor folding and assembly. And third is endothelial nitric oxide synthase folding. How does HSP90 work? Well, this is a schematic of its structure. Again, it has 4 different domains. It has an N-terminal domain, a middle domain, a C-terminal domain, and also a linker domain that's highly charged. And this is very variable. It's not present in all HSP90s. Like for the other chaperones, cycles of ATP binding and hydrolysis cause conformational changes between different domains of HSP90, which then in turn drives substrate binding and release and ultimately leads to folding. The crystal structures of HSP90 reveal the mechanism of action. You can see that upon ATP binding and hydrolysis, there's a very large conformational change. So HSP90 is dimeric, it dimerizes through its C-terminal domain, but upon ATP binding, the N-terminal domains also dimerize, and so it forms this ring-like structure. An opening and closing of this ring structure is what drives the conformational changes and the so-called remodeling or folding and assembly of HSP90's client substrates. I mentioned that by aiding folding assembly, HSP90 can suppress mutant phenotypes, and the emergence of drug-resistant phenotypes is a real problem in drug discovery. In particular, there have been concerns that molecularly targeted therapies are doomed to failure, with resistant clones emerging in response to the selective pressure of any drug treatment. However, a number of recent studies have shown that the evolution of drug resistance in mouse models of human cancer can be restrained by co-administration of small molecule inhibitors of HSP90. Some examples are shown here. So on the left, the example is estrogen receptor, a very well-known substrate of HSP90. HSP90 is important for estrogen binding and for ER activation and stability. And combining an HSP90 inhibitor with the ER antagonist tamoxifen prevents estrogen binding and promotes ER degradation. And this works, results in prolonged anti-tumor effect. HSP90 inhibitors have also been shown to block the emergence of drug resistance in other diseases, so in fungal infections, and to have resistance-free antiviral and antimalarial activity. So there's a lot of interest in using HSP90 inhibitors in combination with other drugs. Lastly, I want to mention proteome organization at the top here. So we discussed how chaperones like HSP90 ensure accurate folding and maintain subunits of proteins, for example, kinases and steroid hormone receptors that lack a binding partner in a soluble state. So as the genes for different subunits of multiprotein complexes tend to be on different chromosomes, aneuploidy, shown at the bottom of the figure here, leads to protein imbalance and non-stoichiometric levels of protein expression. And every subunit encoded by an unbalanced chromosome that functions in a protein complex lacks this binding partner and must rely on chaperones to maintain solubility. And if no binding partner is found, it needs cellular protease, which will come on to the next lecture, for its degradation. And this, if this doesn't happen properly, it can lead to an overload of misfolded proteins and an increased burden on the quality control machinery, and can ultimately cause proteotoxic stress. So one example is thalassemia. These are diseases that result when one or more of the alleles encoding alpha and beta globin is deficient, resulting in imbalanced synthesis of hemoglobin subunits. Proteins that lack binding partners, as well as proteins that fail to get to their correct cellular destinations due to inefficient trafficking, are sometimes referred to as orphan proteins. And this subject is really nicely reviewed in this paper by the Hegder group here in Cambridge. So 2 more points to finish with about protein folding. The first is, so far we've talked predominantly about soluble protein biosynthesis, but membrane proteins are an important class of drug discovery, and they're very different from soluble proteins. They are effectively inside out relative to water soluble proteins. The hydrophobic residues need to be on the outside when they reach their final membrane destination, but before they get there, those hydrophobic residues need to be protected to stop them from misfolding and aggregating in the cytoplasm. And there are a whole host of membrane protein-specific molecular chaperones that do this job. And actually, we know a lot less about the folding mechanisms of membrane proteins because they are hard to study in vitro. They're not soluble, and it's very difficult to mimic the membranes in vitro. So we know a lot less about their folding pathways, but their folding mechanisms do differ and they can go wrong in disease. And one very important example is cystic fibrosis. So CFTR folding doesn't occur properly in cystic fibrosis, and we'll discuss this in lecture 3. You'll already have heard about it from Dr. McVinish's lectures. The second point is that it's now recognized that many proteins don't fit into the conventional structure function paradigm. That is, that if a protein doesn't have a structure, it doesn't have a function. It's now known that around 50% of eukaryotic proteins contain disordered domains, as shown here at the top. These are sometimes known as natively unfolded, and they lack well-defined tertiary structure. And they include many transcription factors, cell cycle regulators, and they're highly represented in disease processes. And examples include MYC, and P53 and BRCA1, involved in cancer, and alpha-synuclein and A-beta, which are proteins associated with neurodegenerative diseases. And we'll discuss both types of proteins in lectures 3 and 4. We can often tell them from the sequences that they are globular proteins. They have very different amino acid compositions. They tend to have very few hydrophobic residues, and they also tend to have low complexity, i.e. lots of the same amino acids. And interestingly, though the cell's quality control machinery has been evolved to recognize proteins that aren't folded, these intrinsically disordered proteins can avoid being degraded. And it's thought that's because because the cellular machinery isn't very good at processing these types of unusual low complexity sequences. So the degradation machineries and pharmacological intervention shown in this box here will be the subject of lecture 2 tomorrow.